Inside your body right now, cells are dividing. Billions of times every day, your cells copy their genetic code and produce new cells to replace old ones. In the vast majority of cases, this process works with extraordinary precision, faithful, controlled, regulated by systems of biological checks and balances that have been refined over millions of years of evolution. But occasionally, under the influence of genetic damage, chronic inflammation, oxidative stress, hormonal disruption, and environmental triggers, a cell escapes those controls. It begins to divide without permission, without limit, without the biological instructions that tell a normal cell when to stop. That is cancer, at its most fundamental level, a failure of regulation. A cell that has forgotten the rules. And here is what the science of the past 2 decades has made increasingly and powerfully clear. The environment inside your body, the metabolic, inflammatory, hormonal, and immunological landscape that every one of your cells lives in, is profoundly shaped by what you eat. Certain foods create conditions that cancer cells thrive in. They fuel the inflammation that promotes tumor growth. They spike the hormones that feed cancer cell proliferation. They suppress the immune surveillance that would otherwise detect and destroy abnormal cells before they multiply. And certain foods do the opposite. They starve cancer cells of the metabolic fuel they depend on. They activate the tumor-suppressing genes that keep cell division under control. They stimulate the immune cells whose job is to identify and eliminate cancerous threats. They reduce the inflammation that cancer exploits as a growth environment. In this video, we are going to cover both sides of that equation, with scientific evidence, with specific mechanisms, and with the practical clarity that allows you to make choices that meaningfully reduce your cancer risk starting from today. This is not about fear, it is about knowledge. And knowledge in this context is one of the most powerful protective tools available to you. Before we go any further, there is something important that needs to be said clearly and honestly. No food cures cancer. No food on its own prevents cancer with certainty. Cancer is a complex family of over 200 distinct diseases, each with its own biology, its own genetic drivers, and its own relationship with the body's environment. The relationship between diet and cancer is real, meaningful, and increasingly well understood, but it is probabilistic, not absolute. What the science tells us is that dietary choices influence cancer risk, that certain foods and dietary patterns are associated with meaningfully higher or lower rates of specific cancers across large populations, that the biological mechanisms through which food affects cancer cell behavior are increasingly well characterized, and that the World Health Organization, the American Cancer Society, the World Cancer Research Fund, and virtually every major cancer research institution in the world now includes dietary guidance as a core component of cancer prevention recommendations. This video is based on that evidence. It is presented to inform, to empower, and to support better dietary decisions, not to replace medical care, not to offer false hope, and not to suggest that diet alone is sufficient protection against cancer. If you have been diagnosed with cancer, please work with your oncology team on your nutritional approach. If you have concerns about your cancer risk, please discuss them with your physician. With that said, let us talk about the science, because it is genuinely important, and you deserve to know it. Cancer-feeding food. 1. Refined sugar and high fructose corn syrup. We begin with the food that, at the cellular level, most directly fuels cancer cell growth, and whose relationship with cancer is grounded in one of the most fundamental discoveries in cancer biology: refined sugar, and its most concentrated industrial form, high fructose corn syrup. In 1924, the German biochemist Otto Warburg made a discovery that would eventually win him the Nobel Prize, and that continues to reshape our understanding of cancer metabolism nearly a century later. Warburg observed that cancer cells consume glucose at a rate 10 to 100 times higher than normal cells, even in the presence of adequate oxygen. Normal cells generate energy efficiently through oxidative phosphorylation, a process that uses oxygen to extract maximum energy from glucose. Cancer cells, by contrast, preferentially use a far less efficient anaerobic pathway, fermenting glucose into lactate even when oxygen is available. This phenomenon, now called the Warburg effect, means that cancer cells are extraordinarily glucose-dependent. They have hijacked the body's glucose supply as their primary fuel source, and they compete aggressively for it. PET scans, the imaging technology used in cancer diagnosis and staging, exploit this directly. They inject radioactive glucose and identify cancer cells by the intensity with which they light up, consuming the labeled glucose at rates that reveal their presence even before structural changes are visible. What this means practically is that a diet chronically high in refined sugar and high fructose corn syrup creates elevated blood glucose levels that provide exactly the fuel environment cancer cells are metabolically designed to exploit. Chronically elevated insulin, the body's response to high blood sugar, compounds this effect, since insulin and its related growth factor IGF-1 are potent stimulators of cancer cell proliferation, signaling cells to grow, divide, and resist the programmed cell death that would normally eliminate abnormal cells. A comprehensive meta-analysis published in the British Journal of Cancer found that high glycemic index dietary patterns were associated with significantly elevated risks of colorectal, endometrial, and breast cancer. Research from the University of Texas MD Anderson Cancer Center found that fructose specifically, the form of sugar dominant in high fructose corn syrup, promoted pancreatic cancer cell growth through pathways distinct from those of glucose, suggesting that different sugars carry specific cancer-promoting risks beyond their shared glycemic impact. The practical implication is clear and well supported. Reducing refined sugar and high fructose corn syrup found in sodas, sweetened beverages, commercial baked goods, breakfast cereals, candy, and the majority of ultra-processed foods is one of the most evidence-based dietary steps available for reducing the metabolic environment that cancer cells depend on. Cancer-feeding food 2, processed meats. In 2015, the World Health Organization's International Agency for Research on Cancer made an announcement that made headlines around the world, and that remains one of the most important and most frequently ignored findings in cancer prevention science. Processed meats, bacon, hot dogs, sausages, ham, salami, pepperoni, and other cured and preserved meat products were formally classified as Group 1 carcinogens. Group 1 is the highest classification in the IARC system. It means that the evidence that these substances cause cancer in humans is convincing and sufficient. It places processed meat in the same carcinogenic category as tobacco smoke and asbestos, not necessarily at the same level of risk, but at the same level of scientific certainty about the causal relationship. The primary cancer associated with processed meat consumption is colorectal cancer, the third most common cancer worldwide. The ARC analysis, based on a review of over 800 studies, concluded that each 50-gram daily serving of processed meat, approximately 2 strips of bacon or 1 hot dog, was associated with an 18% increase in the risk of colorectal cancer. For a disease that kills over 900,000 people per year globally, an 18% increase per daily serving is an epidemiologically enormous effect. The mechanisms are multiple and well characterized. Processed meats are preserved with nitrates and nitrites, which react with the amines in meat proteins during cooking and digestion to form nitrosamines, among the most potent dietary carcinogens identified in research. The heme iron in red and processed meat catalyzes the formation of additional N-nitroso compounds in the colon. The smoking and high-temperature processing of many processed meats generates polycyclic aromatic hydrocarbons and heterocyclic amines, compounds that directly damage DNA in intestinal cells, initiating the mutations that can progress to cancer. Additionally, processed meats are high in saturated fat and salt, both of which promote the chronic inflammation in the colon that creates a permissive environment for cancer cell development and progression. The science here is not ambiguous. The evidence is not preliminary. Minimizing or eliminating processed meats from the diet is one of the single most evidence-based steps a person can take to reduce their cancer risk. Cancer-feeding food. 3. Ultra-processed foods and industrial seed oils. The third category of cancer-feeding foods is the one most pervasive in modern diets, and the one most people consume daily without connecting it to cancer risk. Ultra-processed foods and the industrial seed oils, soybean oil, corn oil, sunflower oil, canola oil, cottonseed oil, that are used to manufacture them and that dominate the cooking oils of fast food and restaurant preparation. The connection between these foods and cancer operates primarily through chronic inflammation, now understood by cancer researchers to be not just a consequence of tumor growth, but an active enabler of it. The tumor microenvironment, the complex of cells, blood vessels, and signaling molecules surrounding a growing cancer, is fundamentally an inflammatory environment. Inflammatory cytokines promote angiogenesis, the formation of new blood vessels that feed the tumor. They suppress the immune cells that would otherwise attack the cancer. They promote the epithelial to mesenchymal transition that allows cancer cells to invade surrounding tissue and metastasize. Chronic dietary inflammation does not cause cancer directly, but it creates and sustains the conditions in which cancer thrives. Industrial seed oils are extraordinarily high in omega-6 polyunsaturated fatty acids, particularly linoleic acid. When consumed in the quantities present in ultra-processed foods and restaurant cooking, these omega-6 fats dramatically skew the body's omega-6 to omega-3 ratio, from the healthy 2-to-1 ratio that human physiology evolved with, to the 15-to-1 or 20-to-1 ratio typical of modern Western diets. This imbalance promotes the production of pro-inflammatory prostaglandins and leukotrienes, signaling molecules that sustain chronic low-grade inflammation throughout the body. Research published in the journal Cancer Epidemiology, Biomarkers and Prevention found that higher linoleic acid intake was associated with elevated risks of breast and prostate cancer. Multiple studies have found associations between ultra-processed food consumption and higher rates of overall cancer incidence, including a large French cohort study of over 100,000 adults that found a 10% increase in the proportion of ultra-processed food in the diet was associated with a 12% increase in cancer risk overall. The additives present in ultra-processed foods, emulsifiers, artificial sweeteners, preservatives, and colorings, are increasingly implicated in disruption of the gut microbiome in ways that reduce its cancer protective functions, producing the short-chain fatty acids that protect colonic epithelial cells, regulating immune surveillance, and maintaining the intestinal barrier integrity that prevents inflammatory bacterial toxins from entering the circulation. Cancer-feeding food 4, alcohol. The fourth food that feeds cancer cells is one that will surprise many people who consider themselves to be following a generally healthy lifestyle. Alcohol, in any amount, of any type, like processed meats, alcohol is classified by the International Agency for Research on Cancer as a Group 1 carcinogen, meaning the evidence that it causes cancer in humans is unambiguous and sufficient. Alcohol consumption is causally linked to cancers of the mouth, throat, esophagus, larynx, liver, colon, rectum, and breast. Globally, alcohol is estimated to be responsible for approximately 740,000 new cancer cases every year, representing nearly 4% of all new cancers diagnosed worldwide. The mechanisms through which alcohol promotes cancer are multiple. When alcohol is metabolized in the body, it is converted first into acetaldehyde, a compound that directly damages DNA by forming adducts with DNA bases, causing mutations that can initiate cancer. Acetaldehyde also impairs the DNA repair mechanisms that would otherwise correct these mutations before they propagate. This DNA damaging effect occurs in every tissue through which alcohol passes, explaining why cancers associated with alcohol span from the mouth to the colon. Alcohol also promotes cancer through its effects on estrogen. Alcohol increases circulating estrogen levels, a direct mechanism through which it elevates breast cancer risk, since estrogen is a primary driver of hormone receptor-positive breast cancer cell proliferation. Research has found that even one alcoholic drink per day is associated with a meaningful increase in breast cancer risk, a finding that has been replicated across dozens of studies in multiple countries. Alcohol generates reactive oxygen species that promote oxidative DNA damage. It impairs the absorption of folate, a B vitamin that plays a critical role in DNA synthesis and repair, whose deficiency directly increases cancer risk. And it suppresses immune function, reducing the body's capacity for cancer cell surveillance and elimination. The important and sobering truth is that there is no proven safe threshold for alcohol consumption in terms of cancer risk. The risk begins with the first drink and rises linearly with consumption. This is not a message designed to create guilt or to demand abstinence from every listener, but it is a truth that the science requires us to state honestly and clearly. Cancer-fighting food 1, cruciferous vegetables, sulforaphane and the cancer switch. The first and perhaps most extensively researched category of cancer-fighting foods is the cruciferous vegetable family: broccoli, cauliflower, Brussels sprouts, cabbage, kale, arugula, bok choy, and watercress. These vegetables contain a unique class of compounds called glucosinolates, sulfur-containing molecules that, when the plant tissue is chopped, chewed, or blended, come into contact with an enzyme called myrosinase and are converted into a family of highly bioactive compounds. The most studied of these is sulforaphane, and the research surrounding it represents some of the most compelling evidence in the entire field of dietary cancer prevention. Sulforaphane works against cancer through a remarkable set of mechanisms that operate at the most fundamental level of cellular biology. It activates a master regulatory protein called NRF2, sometimes called the guardian of the genome, which switches on a battery of over 200 genes involved in antioxidant defense, detoxification of carcinogens, DNA repair, and anti-inflammatory response. In this way, sulforaphane does not simply neutralize a single carcinogenic threat, it upgrades the entire cellular defense system. At the same time, sulforaphane inhibits a protein called NF-κB, the master switch of inflammation that, as we discussed in the cancer-feeding foods section, creates the inflammatory tumor microenvironment that cancer exploits. By simultaneously activating NRF2 and inhibiting NF-κB, sulforaphane attacks the two most fundamental biological conditions that enable cancer to develop and spread. Sulforaphane also directly targets cancer stem cells, a subpopulation of particularly resilient cancer cells that are resistant to conventional chemotherapy and radiation, and are primarily responsible for cancer recurrence after treatment. Research from the University of Michigan found that sulforaphane selectively eliminated breast cancer stem cells while leaving normal breast cells unharmed, a degree of selectivity that is extraordinarily difficult to achieve with pharmaceutical agents. The clinical and epidemiological evidence is substantial. A meta-analysis of 24 studies found that high cruciferous vegetable intake was associated with a 17% reduction in overall cancer risk. Individual studies have found particularly strong protective associations for lung, colorectal, breast, prostate, and bladder cancers. Broccoli sprouts deserve special mention. 3-day-old broccoli sprouts contain between 20 and 100 times more sulforaphane than mature broccoli, making them one of the most concentrated dietary sources of this compound available. A small portion of broccoli sprouts added to a salad or sandwich delivers a sulforaphane dose equivalent to a very large serving of mature broccoli. To maximize sulforaphane release, chop or chew cruciferous vegetables thoroughly before cooking or eat them raw. Light steaming preserves more myrosinase activity than boiling. Adding a small amount of mustard seed, which contains myrosinase, to cooked cruciferous vegetables restores the enzyme activity lost during heating. Cancer-fighting food 2, berries, ellagic acid, and the angiogenesis blocker. The second category of cancer-fighting foods is one that has appeared throughout this channel's content in multiple health contexts, and whose cancer protective properties are among the most richly documented in nutritional oncology research. Berries. Blueberries, strawberries, raspberries, blackberries, and in particular, black raspberries, whose cancer-fighting phytochemical profile is among the most potent of any commonly consumed fruit. Berries fight cancer through several complementary mechanisms. The first and most striking is anti-angiogenesis, the inhibition of the new blood vessel formation that tumors depend on for their growth and survival. A growing tumor cannot expand beyond a few millimeters in diameter without recruiting new blood vessels to supply it with oxygen and nutrients. This process in tumor angiogenesis is driven by signaling molecules including VEGF, or vascular endothelial growth factor. Blocking VEGF signaling is, in fact, the mechanism of action of several approved cancer drugs. The ellagic acid and anthocyanins in berries have been shown in multiple studies to inhibit VEGF signaling and suppress tumor angiogenesis, effectively cutting off the blood supply that a growing tumor needs to progress. Research published in the Journal of Agricultural and Food Chemistry found that black raspberry extracts significantly inhibited angiogenesis in experimental models, with effects comparable in mechanism to pharmaceutical anti-angiogenic agents. Berries also contain pterostilbene, a compound closely related to resveratrol, and a range of anthocyanins that activate tumor suppressor genes. Tumor suppressor genes, including p53, often called the guardian of the genome, are the cellular brakes that detect DNA damage and either initiate repair or trigger apoptosis, programmed cell death, in cells that cannot be repaired. In many cancers, these genes are silenced through a process called methylation. Berry compounds have been shown to demethylate and reactivate silenced tumor suppressor genes, essentially switching the cancer brakes back on. A landmark clinical trial at Ohio State University tested the effect of freeze-dried black raspberry powder on precancerous lesions of the colon in human subjects. After just 9 months of consumption, participants showed measurable reductions in the number and size of precancerous polyps, along with reactivation of tumor suppressor genes that had been epigenetically silenced, a finding that demonstrated a direct anti-cancer effect of berry consumption in human tissue at a clinically relevant dose. Cancer-fighting food 3, turmeric and curcumin, the most studied anti-cancer spice. No single dietary compound has been the subject of more cancer research than curcumin, the primary bioactive polyphenol in turmeric, the golden spice that has been used in Ayurvedic and traditional Chinese medicine for over 4,000 years. As of the most recent literature reviews, over 3,000 published studies have examined curcumin's effects on cancer biology. The findings are consistently remarkable across an extraordinary range of cancer types, including breast, colon, prostate, lung, pancreatic, and brain cancers. Curcumin attacks cancer through a breadth of simultaneous mechanisms that is virtually unmatched by any other single dietary compound. It inhibits NF-κB, the master inflammatory switch, reducing the chronic inflammation that enables tumor growth. It activates NRF2, the cellular defense master switch also activated by sulforaphane, upregulating antioxidant and detoxification pathways. It promotes apoptosis in cancer cells while leaving normal cells unharmed, a selectivity that reflects cancer cells' greater dependence on the signaling pathways curcumin disrupts. It inhibits cancer cell invasion and metastasis by suppressing the matrix metalloproteinases that allow cancer cells to break through tissue boundaries. And it sensitizes cancer cells to chemotherapy and radiation, meaning it may enhance the effectiveness of conventional cancer treatments. Epidemiological support comes from population data showing that India, where turmeric is consumed daily in the diet, has dramatically lower rates of several cancers most prevalent in Western countries, including colon, breast, prostate, and lung cancer. While many factors contribute to these differences, the consistent association has motivated decades of research into curcumin's specific mechanisms. The most significant practical limitation of curcumin is its poor bioavailability. It is not efficiently absorbed from the digestive tract into the bloodstream when consumed alone. The solution is piperine, the bioactive compound in black pepper, which has been shown to increase curcumin bioavailability by up to 2,000% by inhibiting the enzymes that metabolize it in the intestine and liver. Consuming turmeric with black pepper is therefore not a culinary choice. It is a biochemical necessity for maximizing curcumin's systemic effects. Consuming turmeric with a source of healthy fat, such as olive oil or coconut milk, further enhances absorption, as curcumin is fat-soluble. 1 to 2 teaspoons of turmeric powder with a generous grind of black pepper incorporated into a daily meal represents a practical and evidence-supported approach to accessing curcumin's cancer-protective properties through diet. Cancer-fighting food 4, green tea, EGCG and the cancer cell terminator. The fourth cancer-fighting food brings us to one of the most ancient and most globally consumed beverages in human history, and one whose anti-cancer properties are supported by some of the most compelling evidence in the nutritional oncology literature. Green tea, and specifically the extraordinary polyphenol it contains in highest concentration, epigallocatechin 3 gallate, known as EGCG. EGCG is one of the most potent and most broadly active anti-cancer compounds identified in dietary sources. Its mechanisms of action against cancer cells are numerous, well-characterized, and operate across the full spectrum of cancer development, from the initial genetic mutations that initiate cancer through the inflammatory signaling that promotes early tumor growth to the angiogenesis and invasive processes that allow advanced cancers to spread. At the molecular level, EGCG inhibits multiple kinases, enzymes that drive cancer cell proliferation signals, simultaneously. Most targeted anti-cancer drugs inhibit a single kinase in cancer cells, frequently develop resistance by activating alternative pathways. EGCG's ability to target multiple kinase pathways simultaneously makes it more difficult for cancer cells to evade its effects through the genetic adaptations that drive drug resistance. EGCG also activates p53, the tumor suppressor protein that triggers apoptosis in damaged cells. In cancer cells that have retained functional p53, it inhibits telomerase, the enzyme that allows cancer cells to divide indefinitely by preventing their telomeres from shortening, thereby limiting the replicative immortality that is a hallmark of cancer. And it has been shown to inhibit the PI3K/AKT-mTOR signaling pathway, one of the most commonly activated pathways in human cancers and a primary driver of cancer cell survival, growth, and treatment resistance. The epidemiological evidence from Japan, where green tea consumption is among the highest in the world, is compelling. Multiple large prospective studies in Japanese populations have found significant inverse associations between green tea consumption and the risk of breast, prostate, colorectal, and gastric cancers. A study of over 40,000 Japanese adults found that those who consumed 5 or more cups of green tea per day had significantly lower cancer mortality rates than those who consumed less than 1 cup per day. 3 to 5 cups of high-quality green tea daily, brewed from loose leaf or whole leaf tea bags at temperatures below boiling, which preserves EGCG content better than boiling water, represents the dose most associated with protective effects in the epidemiological research. Matcha, a powdered whole leaf green tea, contains significantly higher EGCG concentrations than steeped green tea, as the entire leaf is consumed rather than just the brewed extract. Cancer is not random? Not entirely. Not for most people. The World Cancer Research Fund estimates that between 30 and 50% of all cancers are preventable through lifestyle changes, with diet, physical activity, weight management, and alcohol reduction being the most powerful modifiable factors. That is not a small number. That is millions of cancers every year that the research tells us do not have to happen. The conversation we have had today is part of that prevention story. Not the whole story. Genetics matter, environment matters, chance matters. But diet is a chapter that every person has the ability to write differently, starting from the next meal they prepare. Reduce the sugar, eliminate the processed meats, move away from ultra-processed foods and industrial seed oils, reconsider alcohol with honest eyes, and bring cruciferous vegetables to your table regularly. Eat berries every day. Add turmeric and black pepper to your cooking. Drink green tea in the morning and the afternoon. These are not dramatic sacrifices. They are not expensive interventions. They are food choices, available to almost everyone, supported by decades of serious science, and backed by the consensus of the world's leading cancer research institutions. Your plate is not a guarantee, but it is one of the most powerful tools you have. Use it with knowledge. Use it with intention. Use it in the service of the longest, healthiest life that is possible for you. If this video gave you something valuable, a mechanism you had not understood before, a food you are ready to add or remove, a clarity that feels actionable, please share it with someone you care about. Cancer touches almost every family. This information belongs to everyone. Subscribe to this channel for more evidence-based health content every week. Leave a comment below. Which of these foods surprised you most? Which change are you making first? Take care of yourself, eat with intention, and we will see you in the next video.